She developed the Center for Personalized Medicine at University of Colorado and was the founding head of the Division of Biomedical Informatics and Personalized Medicine in the Department of Medicine there. And University of Colorado was really a leader in this area because of her and her leadership. Prior to that, she was at Johns Hopkins University, where she led a genomics core and a number of NIH-funded programs, working, as a neurologist from the, I have to continue recording, what I'll call pulmonary medicine, but really asthma, immunology, allergy, and really done some really fantastic work in that area. What I found really most fascinating about her CV, other than a lot of really interesting papers, was her Ph.D. thesis, which was in, I'm going to find it here, in the Department of Anthropology. And she did a Ph.D. in biomedical anthropology with the title of The Role of Household Pests in the Epidemiologic Transition of Allergy. And I think the work took place in Barbados, but absolutely a fascinating area. And the reason I bring that up is because not only is it super interesting, because allergy and immunology and asthma is one area where gene environment is really spectacular. In fact, there's some genetic risks that are opposing depending on early environmental exposures. And so it should really be, we gain a lot of lessons from this field. And her work has been spectacular. And she's also an amazing kind of organizer and leader as well. So it's great to welcome you here. Thanks for coming.
Thank you so much. That was an incredible introduction. And Dan, thank you so much. It's great to be here. Sorry for the technical delays. And I apologize if I run over, but yeah, without further ado, it's great to come here and tell my story from the University of Colorado. Several years ago now, I was connected with Clara, which is both personal and professional for me. And I think the similarities between our two institutions and our goals, we are truly the newest kids on the block in the academic personalized medicine space. I think we're very like-minded. And as I'll go through this, I think already we're demonstrating work that we're doing together. A quick disclosure, Dan already mentioned, I just recently transitioned 80% of my effort to Tempus. But what I'm presenting to you today is our academic efforts at the University of Colorado. And without further ado, for those who don't know, population genetics, statistical genetics, genetic epidemiology, pharmacogenetics, et cetera. In short order, we were able to develop a division that's something like 15 primary and 24 secondary faculty, which we're very proud of. As I mentioned, the Health Data Warehouse, what we call Health Data Compass, was launched the year before I arrived. After I arrived, we were able to do something really novel with Google, and we were established a banner partnership with Google Cloud in moving all of our EHR and the various data pieces into the cloud. And I'll talk a little bit more about that. This is old news now because we have in our center moved entirely to the cloud, including the genomics and multi-omics data. But at the time in 2015, geneticists were still wrestling with security around cloud computing for genomics data. So we launched a high performance computing cluster at the time. And then we had to make a decision as to what kind of omics data we wanted to generate. And I'll tell the story in a little more detail, but we landed on Illumina's customized mega chip, and I'll talk more about that. And the goal was to ultimately genotype that on all of our participants. We made a decision very early on the biobank that we wanted it to be a CLIA, ultimately CAP certified biobank. And the reason for that is because our goal was really a hybrid model of research clinical. So by maintaining clinical custody of samples coming into a clinical lab, we would ultimately be able to return clinically actionable results without having to recollect or validate. But it still enabled us in the research space to share research level data. We did something also novel. This was in very close partnership with UC Health, particularly the CIO of the institution. But we were able to launch our consent process through the patient portal called My Health Connection. I'll talk a little bit about that. And that was really a game changer for us as well, moving away from the old-fashioned paper consent, which I did for 20 plus years of my life in genetic epidemiology, to a system-wide electronic consent process. Our primary goal in return of results, we refer to this, and I get in trouble with my pharmaceutical friends and calling it the low-hanging fruit, but truly with pharmacogenomics, it's a relatively easy lift in terms of clinically actionable results that you can return to patients. That said, we, like your program, the IPH, also joined, began partnering with Regeneron about a year ago with the goal of generating research level exomes across our biobank, and then ultimately validating those results to return to participants. So put it all together, and the center looks something like this. We're, I think, similar to UCLA, we're at just around 2.5 million active patients in the UCL system. This is the adult hospital. I mentioned the health data warehouse. That includes actually both UC Health Adult as well as the Children's Hospital. On the left side of the circle, tapping into the EHR, in our case, it's Epic. I think similar to here, the data warehouse, we have just under 9 million patient records in the health data warehouse. On the right side, I mentioned the biobank. We're just under 200,000 consented participants to date, and we've run the mega chip on the first 34,000. I mentioned Regeneron. We entered into this partnership. On the left side, just the talking point that we also entered into in a partnership with Regeneron about 1.5 years ago, again, research level exomes. This image is summarizing the health data warehouse, Compass. And on the left side here, this is to really underscore that Health Data Compass is comprised of clinical data sources, as I mentioned, both of the hospitals, as well as publicly available data. You see quite a variety of data. We have state mortality data, all claims data. That's then combined with these various platforms for extracting data and data analytics. And this just builds out that image, bifurcating, if you will, on the left side, the clinical data delivery versus the research data delivery. So on the left side, if you look at the center, the genomic data that we're generating from the biobank and our clinical biobank, off to the left, then this filters down into the Epic EHR, building out return of results. I'll talk a little bit about clinical decision support, CDS and PGX. We opted to partner with BC Platforms. I had partnered previously with them at Johns Hopkins University. I'm trying to go back to that slide. BC Platforms has a nifty data integration platform. And as you can see from this slide, we leverage that tool both for the return of the clinical data as well as the research data. Basically walks you through the process that we've created, thank you, where the patient electronically reads and signs the consent. This signature in the patient portal then triggers an order for a dedicated blood draw. So the next time the patient comes in for any kind of blood work, a dedicated tube is collected. We maintain clinical custody of that tube. It gets sent to the biobank. Eventually DNA is extracted, and then the goal is 4 and 5, where we generate omics data and ultimately return of results. There are lots of lessons to be learned in building a program, and I remember Clara sharing some of your experiences. We learned a lot from other programs like Geisinger and Mount Sinai, and I think the words of wisdom I was told in 2015 that I didn't listen to is build your unified consent from day one. If your goal is ultimately to return clinically actionable results, we couldn't make that promise in 2015, so ethically we weren't able to create that sort of consent. But for the first just over 100,000 subjects recruited before October of 2019, we went through a primary consent that was for research only, and then added a secondary consent that says, if we generate clinically actionable results, can we return them to you? In retrospect, Of course, we all wish we would have gone to the unified consent. So as of October 2019, we settled on a unified consent, and really it's an all or nothing. If you consent for the research, you're also consenting for return of clinically actionable results and vice versa. The next slide, the next couple slides are screenshots of what that process looks like. And this is, again, the patient portal through Epic called My Health Connection. In this case, Jane has logged on, and she's given an option that she's alerted that there are several consents, and one of the consents is for the CCPM biobank consent. Once the patient clicks on the site, there's an informational video that's available that explains what is personalized medicine, what is DNA, and then they can actually view the consent, electronically sign. And as I mentioned, once that signature takes place, it triggers an order for a dedicated tube the next time the patient comes in for a blood draw. We spent a lot of time thinking about participant engagement. There's been running roughly a 1 to 3 blood sample to 3 consents. And the reason for that is, of course, in our health system, we have a lot of patients who may not routinely get blood drawn. They're orthopedic patients, ophthalmology. We recently piloted and have launched a saliva campaign dedicated to those areas where it's unlikely for patients to have a blood draw. And then we spend a lot of time, we have a participant engagement team that spends a lot of time sending out quarterly newsletters, updates on what's happening in the program, and particularly educating patients if we release, for example, a new drug gene pair, what that drug is for, et cetera. Hopefully this dashboard comes up. Working really closely with UC Health, we put together a monthly dashboard that gets updated, that basically gives us on a monthly basis how many consents have been signed, how many blood samples, and so on. As you can see on the right-hand side, we're also tracking our Regeneron count. Our goal was very lofty at just 450,000 over 5 years. That agreement was executed a week before COVID hit and changed everything in terms of priorities, but we spend a lot of time and effort trying to catch up. So again, this is just sort of a screenshot showing you where we are, particularly down in the bottom middle there, PGX return of results. We keep track of which genes have been genotyped, how many drug gene pairs, and how many patient alerts have taken place through that initiative. So segueing into pharmacogenomics, the next slide is basically illustrating that we think of 3 buckets in terms of clinically actionable results that we would like to be able to return to our patient population: pharmacogenomics, carrier status, and Mendelian disease risk variance. And as I mentioned, pharmacogenomics or pharmacogenetics is arguably one of the lower hanging fruit and impacts a very large number of individuals. So if you look at the literature, a very conservative guess is that roughly 80% of patients would benefit from preemptive pharmacogenomic data in the database. We estimated among our UC Health patient population as well as our biobank population that up to 99% of the population would carry an actionable PGX variant in at least one of the 5 key PGX genes that we set out to tackle initially. CYP2C19 was our first one. That combined with CYP2C9, 2D6, as well as these other 2 genes, could impact up to 99% of the population. So if we just focus on CYP2C19 as an example, we know just from our biobank patients, patient population, that over 50% of those are not CYP2C19 normal metabolizers. So right out of the gate, there's tremendous benefit for more than half of the biobank participants to have these data back in their EHR. And in fact, about 15% of the entire UC Health patient population is on at least one medication that's affected by this gene. If we consider our goal of 500,000 biobank participants over some period of time, almost 43,000 would benefit from having CYP2C19 data alone in their EHR. So I'm going to segue now into the platform of choice that we used in our biobank. And I mentioned already Illumina's megachip. And very briefly, the sort of history behind the megachip and why we were excited about using this platform, also referred to as the Mega EX, is, and this is a busy slide, I know, but if you focus on the bottom, this was really a joint effort in 2014 between the NHGRI PAGE Consortium and our consortium called the Collaborative Study on Genetics of Asthma in African Populations, or CAPA. And what we had done through this NHLBI supported study just prior to 2014, the goal of that study was asthma genetics across the African diaspora, so populations from North, South, Central America, the Caribbean, and Africa. And we wanted to design a genotyping chip that could supplement commercially available GWAS chips, because at the time, the commercially available GWAS chips very poorly captured the diversity in African ancestry populations. With Illumina, we created something called the African Diaspora Power Chip. We took all of that content through this partnership with Illumina to create the mega chip. And in fact, when you look at performance of the mega chip across various ancestry groups, it actually performs best in African ancestry populations, which was a game changer for us. The mega chip was enriched with, it had an exonic backbone as well as clinical content, both PGX, ClinVar, and so on. And so we favored the mega chip as a relatively low cost initial genotyping initiative we could do in the biobank. The next slide just sort of breaks out the different domains or areas, if you will, of the mega chip. So from the mega chip, there's coverage in each of these spaces, as I mentioned, clinically actionable, returnable result, obviously capturing ancestry, GWAS level data from just under 1.8 million SNPs. But importantly, it was also enriched with CPIC and FarmVar content. And so we took it another step. Typically with these Illumina Infineum products, there's headroom for customization. And so what we did as an institution is customize the mega chip by adding CPIC content that wasn't already on the chip. This next slide just illustrates or summarizes for you the various star alleles that were already present on the mega chip, in addition to what we added as an institution in terms of PGX. And then there were several, and of course, this is not a surprise to anyone with copy number variants, and for example, 2D6 that we were not able to capture. So how do we do it as an organization, as a center, to be able to move our PGX initiative forward? We're very fortunate to have two leaders in the space. Chris Acquilante is a PharmD, and Dave Kao, our medical director, form a working group that we named Piccolo. And the goal behind this was to, first of all, construct the technical and clinical informatics infrastructure that we would need to be able to launch a PGX program. And the focus was preemptive genotyping, if you will, and getting this to the EHR. And most importantly, to build the clinical decision support CDS tools that would inform providers, provide the alerts, and make all of this happen. And the next slide is going to summarize for you the various working groups involved in this initiative. The clinical validation working group, that's basically in the lab, taking the mega chip, which is a commercially available research chip. We needed to technically and analytically validate the SNPs for which we were going to return results. So this is an activity largely in the biobank. The tech working group, really the heavy lifters, developing the IT architecture for the actual return of results through Epic, working very closely with them. Of course, the drug gene pair group, making the selections and prioritizing the drug gene pairs. Building out the CDS and integrating that into the EHR is another working group. And really important, if you're going to do this at home, is the education and evaluation working groups. And I'll talk a little bit about why that's so important in just a second here. So the next slide just summarizes that process going from the biobank laboratory, integrating with the BC platforms to get this into the EHR, into, in this case, the UC Health EHR. And importantly, each of these interfaces required, again, development and testing of the results transfer, maintenance of linkage with patient identifiers, and compliance with security protocols, which is a huge lift also. Structured results get transferred into the custom lab records in the EHR. And again, the clinical decision support tools in our case are built directly within the EHR. The next slide gives you a little more detail on this, this process and how we developed it. Evidence review, of course, that's the first step of how we do this, but then the multi-level stakeholder engagement, really one of the most challenging parts of building out a system-wide PGX program. So identifying, for example, the key opinion leaders in each of the clinical spaces, because each of the clinical spaces feels very differently about pharmacogenomics. Identifying what's their preference for CDS design, for example. To give you an example, at UC Health, the cardiologist, they're fine being interrupted. They're fine with a pop-up that makes them go to another screen. Psychiatry wanted nothing to do with interruption. Let me know it's there and I'll decide if I want to use it or not. And that varied also across the entire system. So, our starting point was always the Anschutz campus, but UC Health has hospitals across the state. The other thing we had to be aware of in the CDS development is formularies differ from hospital to hospital. So the alternatives provided, which work in Anschutz, may not work in Fort Collins or Colorado Springs. To give you an example of what these, of what the CDS looks like in real life. So here's an example. Clopidogrel, Plavix was our first rollout, our first drug gene pair, CYP2C19. In this case, there's recommendation that the provider, that pops up to the provider, and then makes the recommendation of alternative drugs to prescribe at that moment. On the other hand, for citalopram, we ended up with this CDS that really makes it optional for the provider to click on the hyperlink that's shown on the bottom, which then takes them to a tip sheet from which they can make their own decision if they want to change the dose or the drug in this particular case. The success of the program, I think, has only recently been recognized system-wide. This is a story that UC Health published a couple months ago, showcasing a particular patient of one of our providers whose nighttime anxiety had increased, and the provider considered upping the dose of citalopram. The pop-up came up that said, hey, this patient is a rapid, ultra-rapid metabolizer. And so it wasn't that the medication was failing, it was just that he metabolized the drug faster than others. And we were able to interview the patient, and the patient said, well, I gave this blood sample a couple years ago, several years ago, for the good of research, and hey, I benefited from it. So I think that's a real testimony to how these programs can work for actual patients. This is just a timeline, if you will. We launched our PGX program in July 2019. As I mentioned, we started with clopidogrel. We're agnostic to the clinical phenotype. So our approach has been get as much mileage out of we can, each, one gene at a time. So you can see we've gotten considerable mileage out of CYP2C19 across different, clinical therapeutic areas. We've just moved on to SLC01B1. And we're really excited. In the pipeline is DPYD, which will be our first oncology PGX drug to release. This story and how we accomplished this is in a publication that came out a little over a year ago. When we look across research biobanks in the US with more than 60,000 samples, and which are returning clinically actual PGX results, We think we've been especially successful in returning what we have so far. What do we attribute this to? First of all, strong institutional support is critical. Genotyping in a CLIA environment has been an advantage for us, allowing us to return in real time. It really takes a multidisciplinary team to succeed in this area with expertise, not just in pharmacology, but in clinical education and so on, and proactive strategies to engage our stakeholders. And unfortunately, the average provider doesn't have the in-depth genetic background training to understand and feel comfortable with making these decisions. So there's just a lot of education and evaluation that has to take place. So again, it's summarized in this manuscript that we came out with. So lessons learned. First of all, unified consent model is ideal. Wish we would have done that from the beginning. What I haven't interjected is that we have been going through a campaign of reconsenting. So for any individual who signed a previous not unified consent, they get a pop-up whenever they log into their patient portal that says, hey, do you mind signing this consent again? And this is why. I mentioned the strong institutional support, in our case, especially from the CIO and IT services. And our philosophy is one gene, many drugs, as I already mentioned, and one therapeutic area, many genes approach just wasn't possible using the platform that we did. We feel at the end of the day, we played to the strength of the chip that we used. And I've already mentioned that workflows and formularies vary across the health system, across different hospitals, and that's a really important consideration. In terms of other lessons learned from this experience, I would say consistent messaging and myth busting, we call, is key. So for example, PGX versus disease genetics, there's a lot of confusion over PGX and how that's returned versus incidental findings, for example. The advantage of PGX is, we think of it as akin to saying you're allergic to penicillin, which is very different than saying, hey, you have an incidental finding, you're at 80% chance of developing breast cancer, ovarian cancer, prostate cancer, which requires a completely different layer of consenting and genetic counseling. Striking that balance between the needs and wants of the clinical subject matter experts, the SMEs, can be tricky. And I've given you two sort of extreme examples of that, but this is a really important consideration. Traditional educational models simply aren't practical. And of course, COVID changed all of that, as we're experiencing right here today. It became much more challenging to have in-services and bringing providers in. So we have to get creative in thinking about how we're educating our SMEs and the providers. So to conclude on the PGX portion, I'll just say, we believe that preemptive clinical pharmacogenomics implementation via a research biobank is feasible. It requires, as I mentioned, this institutional support. If you're already starting in a clinical environment, you've got a leg up in being able to return these results, multidisciplinary and proactive strategies to engage stakeholders. So when we think about what's next for PGX, I've already talked to you about the customized Mega 2. On that chip alone, we have 6 additional genes we're going after with 6 new drugs. This could bring our total number of drugs to 23. On the horizon though, and just being released now, is Illumina's Global Diversity Array, or GDA, with an enhanced PGX booster. And we've spent a lot of time talking with Illumina and considering how we will move to this platform at CCPM. The advantages of this chip over the previous version are tremendous. So what Illumina has done is modified their Infinium chemistry to now capture structural variation, and much of what we couldn't capture previously, for example, in 2D6, are CNVs and so on. And so if you look at the data from this particular chip, and at CCPM, we tend to bias on the CPIC AV guideline, drug gene pairs. There's a total of 444. And I've confirmed with Illumina before this presentation, all of the CPIC AB are completely captured on this new version of the MEGA, the GDA. So that's an exciting space to be. Our program, and it's probably no surprise given my own background and interests, that we've built a program that's very much focused on the role of ancestry and the importance of ancestry in considering clinical implementation of personalized medicine. This is a really nice review paper that came out a couple of years ago by our friends from Mount Sinai, Nora and Emer, which basically showed this progress that we're making from the very first biobank, if we think of Decode Genetics, being exclusively European, and the increase in non-Europeans in national and international biobanks. I've circled Colorado. Again, this was a couple years ago. UCLA, you were already ahead of us 2 years ago when this paper came out. But it's an exciting space to be in because it's really showing that biobanks as a whole, and of course the All of Us program, has added considerably to the space of increasing that diversity. And within CCPM, in fact, we have a unit that focuses on nothing but ancestry, both for research and clinical purposes. It's led by Chris Janou and Jonathan Short. And this illustration simply shows you of the first 500 random subjects from our biobank who were genotyped on the MEGA, what that distribution of ancestry looks like for us in Colorado. This is a challenge, an important area in terms of return of clinically actionable results. If we think about just 3 years ago, PharmVar or PharmGKB, or prior to 2018, excuse me, we relied on these 5 OMB, lump them all in a bucket sort of ideas around ancestry: white, Black, American Indian, Asian, and Native Hawaiian. After July, PharmGKB annotated or relied then on the 1,000 Genomes Phase 3 and HGDP to broaden those categories. And this is really important. And why does this matter? So when we reflect back on, for example, our CAPA program, and this slide is coming up next, in the CAPA project that I mentioned, again, our goal was to characterize the African diaspora genome. We had contributed to Phase 3 of the 1,000 Genomes project. Dan mentioned the Barbados population. Barbados alone was representing all of the Caribbean, and we knew that wasn't accurate. And so through the CAPA program sponsored by NHLBI, which continues now, we sequence up to 1,000 individuals from these 21 different sites to better characterize the African diaspora genome. And we learned several important clinical facts from that exercise. This one paper, the first paper, was led by Tim O'Connor at University of Maryland, which juxtaposed what we learned from the CAPA genome, the African diaspora genome, with what was in ClinVar, showing tremendous missing information in terms of likely pathogenic variants for individuals of African ancestry. And in work that we did in collaboration with Stephen Salzberg at Johns Hopkins, went back and reassembled what we called the pan-genome, pan-African genome, and actually discovered that the pan-genome, the African pan-genome, contained about 10% more DNA than had previously been known in public databases. So why does this matter when we think about a precision medicine program and returning clinically actionable results, even focusing on pharmacogenetics? So what this slide does is illustrate for you across 2D6, 2C9, and yeah, three star alleles for 2C9, and in the upper left, 2D6, and showing you using 1,000 genomes data, the minor allele frequency of these star alleles that we consider CPIC level guideline pharmacogenomic drug gene pair data. And if you look, for example, and this is important for warfarin, in the lower right-hand corner, look at that, if you will, difference in minor allele frequency in individuals of African ancestry compared to, in this case, North and South America. To look at that a little bit closer, this next slide breaks down the distribution of 2C19 star alleles across the different ethnic groups shown on the x-axis. And what you can see, particularly for African individuals, is a really tremendous difference in terms of what are the most relevant, what are the most important star alleles for 2C19 for Africans compared to Europeans and basically everyone else. And so this would typically be missed in a clinical setting without digging deeper into what is most relevant in non-European populations. I want to close with what we kind of see as the next stage in precision medicine, leveraging this technology and preemptive genotyping and hopefully soon sequencing. And that is thinking of the clinical utility of these data vis-a-vis polygenic risk score. So for those who don't know, what is polygenic risk score? I've got one of the leaders in the field sitting in front of me, but for those who are not aware, it's a sort of fortune telling about, predicting your risk of disease for polygenic disease that's not typically thought of as monogenic. It's this idea that a number can be derived from variation across multiple genetic loci and their associations, and it's weighted to create a score. And let me try to bring up this whole image. So a trainee in our group, Chris Earhart, came up with this really nifty illustration that I think pitches it very nicely. It's animated, but I just sort of blasted through it to get it up here. So on the upper left-hand side, we have a GWAS, we have multiple hits, but they get different weight. And we create an estimate based on a particular outcome. This particular example was for atopic dermatitis that we published on recently, of creating a risk score, if you will, for developing atopic dermatitis. For those who are familiar with this trait, filaggrin is a gene that's sort of tried and true and thought to contribute significantly to atopic dermatitis, but not always. And what's interesting about filaggrin is that it actually carries relatively low risk in individuals of African ancestry who suffer disproportionately from atopic dermatitis. So PRS is a really nifty way to get at what is the risk of an infant developing atopic dermatitis. And why does this matter? If you know that an individual, and in this case, a very young individual, is likely to develop a disease, there are proactive therapeutics that the provider could prescribe for an infant to at least minimize the severity of the disease. And we recently published this. And the reason I'm showing you this is because we were able to use as a training set an NIH-sponsored research study that we had conducted for almost 20 years, the ADRN. And then we were able to implement this in the CCPM biobank using the 34,000 data points that we had, in addition to 23andMe and UK Biobank. So it's very powerful. This paper came out literally, I think, a week ago. Pradeep Natarajan at the Broad, and it has this very nice illustration of what is a polygenic risk score, and this is really showing you this distribution of these scores, if you will, across using data from multiple reference populations, but prescribed for a particular individual, and showing in that to the very right there, that liability threshold can be very predictive. And in this image that Pradeep used in this recent review paper, I think is very nice. This is for familial hypertension, or cholesteremia, sorry, hypercholesteremia. Traditionally, we think of variants in the ApoB or the PCSK9 genes as very predictive of developing FH. And what they've been able to demonstrate is that PRS actually goes a step further in providing effect estimates for individuals on the right end of that tail that actually perform better than testing for these monogenic variants alone. And this is just to underscore, and I think I had to miss the conference earlier today because I'm here, but we're very excited to be part of an NHGRI initiative at CCPM, and I have folks listed here. Well, the slide still hasn't come up to all of you, but this is a consortium focused on PRS funded by the NIH, and importantly, really focusing on portability of these scores across different ancestry groups. Of course, you all have the Atlas program that Bogdan's leading. CCPM, Leslie Lang and Chris Chenu are leading it. at CCPM, and then we're taking advantage of some of these existing diverse cohorts to enrich this effort. So to close with is what have we built so far? I'll just summarize by saying, we were able to, in relatively short order, similar to what you all built here through IPH, develop an institution-wide initiative that's already beginning to benefit our patient population. We're one of the first enterprise health data warehouses on a public cloud platform. As I mentioned, we've now moved all of our data, including genomic data, to the Google Cloud. We think our CLIA-CAP biobank is one of its only kind. In addition to the benefit of PGX, which I described, and thinking about the Regeneron project, of course, those exomes are generated in a research laboratory. So when they come to us, there's the challenge of validating clinically. The 3% who are affected if you just focus on the ACMG 74. So we feel we have a little bit of an advantage because we've been able to maintain clinical custody of these samples that we can go back and validate directly on those samples. We're the first biobank to leverage the mega chip, a commercial research chip for return of clinically actionable results. And we think we're one of the first to launch clinical integration of PGX through a research biobank. As I've already mentioned, we're currently scaling up to generate whole exomes on 450,000 participants by 2025, hoping that COVID will settle down so that we can move forward with that. And the next slide is the last slide is just to brag a little bit about our website and how folks can get more involved, how we can collaborate on research initiatives. And I'll end there. And the last slide is a picture of all of us and lots of folks to thank. I really want to shout out to Chris Aquilante and Dave Kao, who have led the PGX initiative. We work very closely with Rasika Matthias at Johns Hopkins, have worked some of the data I showed you also in collaboration with the Mount Sinai team. And then 3 of our big partners, BC Platforms, Illumina, and Google Cloud. So thank you.
Thank you so much. I'm going to try to figure out, number one, not only fantastic talk, but you actually got over all of that technical detail problems and
it kept me awake.
And you did it before the hour was up, which is amazing. So let me see if I can look at questions. Oh, maybe on this one.
Oh, yeah.
Are there any questions from first we'll start with the people who came here in person. Are there any questions there? So we have, Luce.
So when it comes to results, is there any timing
Yeah, so the question is about the timing of return of results. So go for it.
That's a great question, Luce. So it's not something we're especially proud of. If you imagine some of these individuals were recruited, consented several years ago now. So in terms of giving something back we think of incidental findings. So for the example, the Regeneron project, you guys probably are experiencing the same sense. Some of these samples have been in the biobank for say 5 years, even have to be reconsented to return that level of clinically actionable results. And what I didn't mention is we've spent a lot of time in anticipation of the Regeneron exome data, which initially will focus on ACMG 74. In what does that return process look like? It simply isn't scalable to do it the old-fashioned way. And we did a pilot that basically confirmed that our adult medical geneticist plus a counselor can see a maximum of 2 patients a day. That's simply not scalable. We feel we already had a telehealth initiative well underway at the institution. COVID has augmented that. So we're spending a lot of time thinking about how to engage patients for return of incidental findings. For PGX, I see it a little bit differently because preemptive genotyping for return of PGX, you, for example, if you contributed in 2015, your sample, your age, your fitness, you're probably not going to be prescribed clopidogrel for another decade or more, if that, if ever. So in terms of preemptive genotyping for PGX, it could be years before a patient benefits, right? For the incidental findings, that does keep us up at night in terms of thinking about, how to get back to patients and let them know.
I have a related question, which is with these Illumina arrays, especially rare variants, I'm wondering how accurate its rare variant genotyping is, and from a CLIA standpoint, how comfortable you are using the Illumina array, for that level of genotyping when you're giving individual specific genotypes back.
Right, it's a great question, Dan. So there is, and we have not dug into it. I've looked at the evidence, the validation. For the GDA, in addition to the PGX booster, there's a cytogenetics booster and there's a carrier screening booster. We haven't gone that route. And to be honest, probably like you all, we're at least for now going to rely on the exome data and validation of, again, that 3%. For PGX, it's a lift, but again, what we did with the Mega is what we will do with the GDA is the technical analytical validation of those SNPs in-house. But I can say there's quite a bit of data that Illumina will share with you in terms of the validation, and it's pretty good. Yeah.
So good enough for CLIA.
Good enough for CLIA.
Yeah.
Yep. It's so long as you go through the technical analytical validation of that marker on the chip.
So we'll take one more question here because we are a little over, but there's some great questions. And so, okay, we'll take two more. So I'm going to take one from this audience, from the computer audience, which is, what are your plans on combining PRS together with rare variants like BRCA1 breast cancer, and how would this influence screening for breast cancer?
It's a great question. So we believe in the space. I mean, for each of these diseases, breast cancer included, there's a polygenic component. And so if the question is related to how we're proceeding in the center, there's how we would screen, for example, for breast cancer using the exome data that comes back from Regeneron, and then combining, for example, the GDA data to generate predictive algorithms around breast cancer. I can say, wearing my other hat, we're thinking a lot about this and creating platforms that actually can combine an exome backbone, a low pass whole genome sequencing backbone, spiking in additional variants that are poorly captured with those two platforms alone. And I think, again, the familial hypercholesteremia table from that recent review paper on PRS, I think is a great example. Two genes known to be strongly associated, predicting risk of FH, but we know that all FH is not caused just by variants in these two genes, and that's where PRS comes in. So our vision of the future is really combining monogenic with polygenic using these different platforms together.
Great. One last question, and then we'll thank you.
Thanks for an awesome talk. I had a question about whether there's a plan to sort of reanalyze data as more data becomes available. So for example, if a better polygenic score becomes available, or if there's a new clinically actionable variant, I guess, is there a plan to do that? And if so, I guess, what are the implications for disseminating that as well? For example, someone might think, oh, I don't.
So the question is there a plan to reanalyze data, both, I would assume, from the incidental findings as new incidental clinically relevant genes are identified, as well as on the polygenic end as those get better and better?
Yeah, that is a great question, and we wring our hands a lot around that. So for example, just to limit it to the PGX conversation, we went through the process with the mega chips, and each time there's a new lot, you have to revalidate technically, analytically, those variants. And so our lab director in the biobank, we had this agreed upon list for the initial technical analytical validation and then a freeze. So in terms of reanalyzing data from a research commercial chip, that's a problem. You know, our vision in the future, of course, is that every patient who walks through the healthcare system has a clinical genome, and, you know, it's sequence once, test many times philosophy. With the technology we're using right now, we're not there. That's a great question.
Thank you. Well, thank you so much for
Yeah.
putting up with the technical
It's good. All good.
defects that we have here, and really thanks for a great
Thank you,
visionary
Dan.
talk.
It
Yeah.
was great to be here. Thank you.